Hi everyone, I'm Adam Guss from Oak Ridge National Lab. I'm the PI for Oak Ridge National Lab and also a co-lead of the host onboarding team, as Chris just said. Next slide, please. As we think about ways that we can partner with the host development that we're doing, obviously we can offer a lot of basic tools that I'll talk very briefly about on the next slide. Things like transformation methods, promoters, gene knockout systems, and so forth. But we also know that we can really accelerate progress by helping people to develop more advanced tools like CRISPR-Cas editing, recombinering, CRISPR inhibition, and the SAGE toolkit that I'll talk about. And so we have a lot of these tools already in-house. And so adapting them to new organisms can be really, really rapid. And so when you guys come to us with your favorite organism that needs these tools, we can really push forward rapidly on some of the tools that you might need. Next slide, please. So for some of the basic tools, we do things like improve transformation efficiency. We do this by understanding how the cells protect themselves against foreign DNA, and then come up with ways of evading those defense systems to make it easier to get our DNA in. Once we have that done, we can very rapidly characterize promoters, both in aerobic and anaerobic organisms, because we have large collections of different genetic parts. Origins of replication, selectable and counter-selectable markers, reporter genes, promoters, and so forth. And so then we can leverage all of these tools to develop things like homologous recombination tools for gene deletions or for insertion of DNA into the chromosome. Next. Another tool that we develop a lot and adapt to a lot of new organisms is a tool that we call SAGE. And this uses a class of enzymes called large serine recombinases. So a lot of people know about other recombinases like Cre recombinase that works on LOX sites, Flip recombinase that works on FRT sites. So these site-specific recombinases are they're often from phage, and their job is to insert the phage chromosome into the bacterial chromosome. Unlike Cre, LOX, and Flip Frit, these recombinases recognize sequences that are different. So two different sequences that are typically called ATB and ATP. This recombination works really, really efficiently, and it works in a test tube. It works in pretty much all domains of life. So pretty much any organism that's not an extremophile, these enzymes should be able to work in. And they've even been used in plant and animal cell lines and across the microbial domain. So these enzymes are really useful for strain engineering, and I'll show you how. Next slide, please. So there are also a lot of these a lot of different recombinases that can all be orthogonal with each other. And this is where this recombination becomes even more powerful. So what we'll do is we'll start by using traditional tools to insert a landing pad of recombination sites into the chromosome. Then next, we can co-transform two different plasmids, one of which expresses the recombinase transiently, and the other of which has the second recombination site and the genetic cargo that we want to insert into the chromosome. Next, you then get transient expression of the recombinase, which then goes over and inserts your cargo plasmid into the chromosome. This works very, very efficiently. And so it's a very simple and efficient way of inserting DNA into the chromosome. It works just as well as transforming plasmids, but instead of replicating autonomously at multi-copy and all the challenges that come along with that, instead it stably inserts it into the chromosome. Next. And we put this landing pad into lots of different organisms. So we started with Pseudomonas putida, which is a gamma proteobacterium, but we've also put it into Corynebacterium glutamicum, which is an actinomycete, Cupriavitis, which is a beta proteobacterium, Zymomonas, which is an alpha proteobacterium, Rhodococcus, which is an actinomycete. And we've also put it into Clostridia and bacilli. And so this tool allows us to very rapidly put DNA into very diverse organisms. Next. And like I said, it's very efficient. So this is just looking at the efficiency of DNA insertion at each of these different recombination sites in 3 different organisms, a gamma, a beta proteobacterium, and an actinomycete. And over on the right side, you can see the efficiency of homologous recombination and transformation with a replicating plasmid. You see that insertion with SAGE is much more similar to transformation with a replicating plasmid than it is with homologous recombination. Next. And where this gets really powerful is when you start being interested in adapting tools to new organisms. So we put this landing pad into, like I said, Pseudomonas butida, Corynebacterium, and Cupriabitus. And we built a small library of promoters. We were able to transform literally the exact same library into all three organisms. And so if you imagine having a new organism where you want to develop genetic parts, after we get the landing pad in, literally a week later, we have a characterized promoter library. And we can do the same thing with other genetic parts, whether it's different reporter genes or selectable markers or terminators. But we can also do this with pathways. And so you can start to ask questions like, what is the best organism for a pathway, rather than the traditional, what is the best pathway for an organism? Because we can take literally the exact same plasmid or library of plasmids and put it into lots of phylogenetically diverse organisms. Next. We can also use this tool to integrate CRISPR tools. So we've done a lot of work at Oak Ridge and across the ABF with different CRISPR systems, both for genome editing and for CRISPR inhibition, for genome scale, like genome-wide inhibition libraries. We've used Cas9 and Cas12, and we've used native CRISPR systems. So we have lots of experience with diverse CRISPR systems that can suit a variety of different needs. And we can do this in lots and lots of different organisms, which is really enabled by the SAGE system. Next. And then the thing that I think is most cool about SAGE is that in an organism where the transformation efficiency is really high, like Pseudomonas butida, SAGE works so well that we can multiplex insertion of DNA into the chromosome. So we're able to simultaneously insert 3 different plasmids into the chromosome, and so you can start thinking about building combinatorial libraries in your organism of interest. Maybe it's a metabolic pathway where you have a transporter and 2 catabolic genes. You can just transform all of this in and build your library in your organism of interest. And also the organism doesn't know if the DNA is pure or not, so you don't have to put in single plasmids. You can put in libraries of different components to help find the best combination of different genetic components for building your pathway. And then we have the ability to simultaneously remove all 3 selectable markers in a single step. And so then you could do this a second time and insert another 3 plasmids or 3 libraries into the chromosome using the SAGE technique. So next slide. So just very quickly, we've done this in lots of different organisms across the phylogenetic tree of bacteria. And it works really well. We have ongoing work where we're adapting this tool to filamentous fungi and to yeast, and we're adapting it to a variety of bacteria and members of the archaea. Next. And then we also partner with companies to develop both the basic tools and things like SAGE and CRISPR to the different organisms that are of use to our partners. And so if you're interested in working with us to adapt some of these tools or develop even the basic tools that you need for the engineering work that you want to do, please reach out and we're happy to talk about partnering opportunities.